Good morning to all of you. First and foremost, it's a privilege and pleasure to be here in this meeting. Like the director pointed out, it's interdisciplinary. I'm Purusharth Rajyaguru. I'm an associate professor at Department of Biochemistry, IISc Bangalore. My big thank you to Sandeep and Debayan for putting this meeting together. It's indeed my first time to IMSC, and I must say it's a wonderful, quaint little institute in Chennai, and really happy to be here. Yesterday, while sitting at the dinner table, I realized that it's going to be a nice, mm, uh, diverse assembly of course, experimental biologists, but along with that, computational biologists, physicists, chemists, and we all are sitting at the Institute of Mathematical Sciences, right? So, with Science Day around the corner, I think it's a wonderful meeting to have during this time of the year. So, since this is the first talk, and we all come from sort of different backgrounds, I have worked around my talk yesterday night to give a little more elaborate introduction than I would normally give. I still don't know how much is it going to help, but I'll try my best. And I have not tried to keep it very data heavy, although I'm an experimental biologist, so I'm excited about the data that we have, and I would like to share that so that we can get suggestions and have some collaborations. But that's how I'm going to try and do it. But I'll be very happy to take questions, obviously at the end, and also later on in case if I run out of time. All right, so I'll be talking about RNA condensate disassembly. Role of low complexity sequences in biological form and function. This is sort of the big picture thing that we are excited in our lab, right? This is what all the projects more or less try and cater to understanding. Okay, so what are low complexity sequences? Again, I'm sure this is going to be very preliminary for some of you, but I still have it here just in case, there are non-biologists, and if these terms are new to you, then we all could come to the same point before I start talking about the science part. Okay, so these are sequences which are not equally representing all the kinds of amino acids, which is usually the case in most of the proteins, that if they are not equally represented, they're more or less uniformly presented in a given protein sequence, right? But if you look at this sequence, that's really not the case. You can see there is over-representation of some colors and highly underrepresented form of some other colors, right? And that says that the complexity in this sequence is low. You can see lots of repeats of G, lots of polar residues, very few positive residues, very few hydrophobic residues. These kind of sequences are known as low complexity sequences, and they are surprisingly very common in the human genome, right? And people are now warming up to the idea that how could these be functioning? What is the role of these sequences? What is becoming even more clear is that the idea earlier used to be sequence determines structure and structure determines function, right? In biology, at least that's what we have studied in textbooks. But when you look at the human genome, it looks like intrinsically disordered regions, which are the feature of these low complexity sequences. Because they do not have enough information in them, they always do not fold very well, or they often remain in a disordered form. And therefore, they are known as intrinsically disordered sequence, and the proteins that contain them are known as intrinsically disordered proteins, right? So only 37% of the proteins in human genome are fully folded proteins, right? As opposed to that, 58% of the proteins in our genome, they have intrinsically disordered regions, right? So they could have IDRs constituting less than half of the protein, or even more than that, right? And of course, there are 5% which are completely disordered, right? So vast majority are the kind where you have some folded regions in a protein, but you also have these low complexity sequences which cause intrinsic disorder. And this doesn't make much sense, because if we think that structure is so much important to the function, then why has human genome so many proteins that tend to remain disordered, right? And that's one of the basic ideas that we are trying to understand in the lab by focusing on these intrinsically disordered proteins through the low complexity sequences. Okay, so I just mentioned that to you, that this is an important part of the human proteome. These can usually they are disordered, but they can take up certain specific structures under certain conditions, right? So these could be in response to specific solution conditions, in response to binding to specific ligands, or in response to changes in temperature, so on and so forth. And that keeps happening all the time inside a cell, right? I mean, all these sequences are not present in isolation. They keep bumping into other biomolecules. They experience different kind of conditions, including stress. And therefore, it's conceivable that although they do not form structure under specific conditions, but in response to binding to ligands or upon coming other conditions, they'll acquire structure. So they keep going back and forth between a form where they can have a structure, whereas in the other cases, they could be intrinsically disordered. Okay, so specifically, my lab focuses on understanding the role of these sequences in RNA biology. We have had a group at IISc for past 11 years, and we are interested in understanding mRNA fate in the cytoplasm. How does an mRNA get translated? When does it get stored? When does it get degraded? So how are these different fates decided in the cytoplasm? And more specifically, how do these LCS containing sequences, which often have IDRs, contribute to this mRNA fate determination? Okay, so we think that all the mRNAs coming into the cytoplasm get translated. That is not really true. There are lots of mRNAs that come to the cytoplasm and are not getting translated. Instead, what happens is that they often remain stored in the cytoplasm in the form of a translationally repressed state. This is often a global feature of a stress response. So anytime any kind of a cell comes across a stress, the number one response to that stress is downregulation of translation, which is protein synthesis. Why? Because protein synthesis is an extremely expensive process to undergo. And therefore, under stress conditions, you're only thinking about mounting a stress response to that specific condition. You're not worried about your housekeeping genes. So you want to downregulate 95% of the translation so that A, you are conserving energy, and B, you want to focus on synthesizing those proteins that are going to help you mount a stress response, right? So during that times, mRNAs are stored in the cytoplasm, and this storage often occurs in these structures known as RNP condensates, also known as ribonucleoprotein condensates, which are nothing but complex of RNAs, RNA binding proteins, and mRNAs. There could be other RNAs also present in these structures, but from the purpose of my talk, I'll mostly be focusing on mRNAs and mRNA binding proteins. So these condensates assemble, and then often when the stress is gone or when these RNAs need to be translated, they disassemble, which is known as de-repression, because now these mRNAs come out of these condensates and they can start getting translated by associating with the ribosomes. While they are in these structures, they are by and large not getting translated. Okay, I'm using the term by and large because there are also exceptions to that kind of a rule. So largely the idea is that in the condensates, you remain stored. You're not getting translated, you're not getting degraded, but when the stress is gone, you come out of these structures and you can start getting translated, right? So what we try and understand is, how does mRNA get stored? How do these RNP condensates, which can also be referred to by terms as RNA granules, puncta, all these I'm going to be using as synonyms for the term biomolecular condensates or RNP condensates. So don't get confused if I end up using different terms, but they all are RNA protein complexes. And how do mRNAs move out of these condensates, which happens upon disassembly of these condensates, allowing translation? So that's basically what we are excited about in the lab, and almost all projects try and understand that. What are RNP condensates, right? I already told you, but just a formal introduction, that these are conserved membraneless compartments that determine mRNA fate, right? I've kind of explained that in the previous slide. Now I'm just going to show you what they typically look like. So I'm an experimental biologist that works with yeast. For the first 10 years of our lab's existence, we were completely working on yeast. And for past 2 to 3 years, we have organically moved from working from yeast. We continue to work on yeast, but we are also working on human cell systems. And some of the data that I'm going to show you is actually a result of this transition, which has allowed us to explore some things that 11 years ago, I would have never thought that I would be working on these aspects. So that's what I'm excited about sharing today. So this is an RNA binding protein that's stacked to GFP. What you're looking at is a yeast cell. Yeast is a very simple single-celled eukaryote. We are also eukaryotes and so is yeast, but we are multicellular and we have this wonderful body organization. Yeast is a single-cell animal, but the beauty of yeast is that a lot of things that are known to be happening in yeast also seem to be true in you and me, right? So yeast is a wonderful model system to work with because you can apply genetics, biochemistry, cell biology very nicely to this model system, figure out mechanisms, and then take it to the human cells and ask, is it still true in higher organisms? And it turns out that it's indeed true in most of the cases. Now, when this cell is stressed, you can now see that this green color is all, you no longer see the whole cell because the signal is all getting concentrated into these punctas, right? Dots. These are what we call as condensates or RNA granules. So these are the sites where RNA gets stored, and what you're looking at is actually the protein. So there are lots of RNA binding proteins and RNA which are stored in these structures. And this happens when the RNA needs to be stored. You put them back in a happy condition, they'll revert to this. So these are dynamic structures, and this keeps happening all the time in the cell, that the granules or condensates assemble and then they disassemble. Like I said, this is conserved. So it just doesn't happen in case of yeast. It happens in human cells. It happens in, I'm sorry, the names are not really showing up properly. This is, you're looking at what happens in case of C. elegans, where P granules were identified, and there's a lot of work that is being done in the context of P granules. These are human cells. These are actually neurons. So neurons are present in our brains, and they, of course, play a very important role in these neurons. Also, these structures. So it's a very conserved structure that plays an important role. And although there are different names to these structures, but there are two main kinds of RNP condensates, right? There are stress granules and P bodies, which comes from the name processing bodies, right? And both of these are more or less same kind of structures, except that stress granules are only sites of mRNA storage, whereas processing bodies also have some enzymes that can degrade RNA, right? So in P bodies, there is a possibility that some of the stored RNA could get degraded, whereas in stress granules, mRNAs are basically stored and kept until they are released back for translation, right? So these are the two main kinds of RNA granules which are very well characterized. So what you're looking at are different kinds of granules on this slide, but for the purpose of this audience and this talk, I'm not going to make it very complicated. Suffices to say that this is a conserved process that happens from the simplest of eukaryotes, which is yeast, to you and me. Okay, so one of the aims of this meeting is to try and understand different facets of liquid-liquid phase separation. And where does that fit into all that I have been saying? So in 2009, an article by Clifford Brangwine, who at that time was a postdoc with Tony Hyman, and we have people in this room who have connection with Tony Hyman and have undergone training with him, actually demonstrated that these P granules in C. elegans have liquid-like properties. And that was one of the first papers to propose that biological moieties could behave like liquids and they undergo phase transition. This term itself went transition, and now phase transition has become phase separation, and it's mostly referred to as LLPS or liquid-liquid phase separation. But what does that actually mean? All right, this is a very simplistic representation, right? So bear with me. So this assume this is the cytoplasm of any kind of a cell, and you can see there are lots of different proteins, and let's assume for a moment that these are blue colored things are RNAs and the others are other biomolecules. They are present all over the cytoplasm, homogeneously distributed. Now, there is a physiological cue or a stimulus, and as a result, what starts happening is that certain part of the cytoplasm, these undergo a change in concentration. So in some part of the cytoplasm, the concentration goes up. In the other part, the concentration goes down. When the concentration crosses a certain threshold, that's when some of these proteins and RNAs, they start phase separating, right? When it crosses the saturation concentration known as CSAT, that is when they phase out of the solution and they form a moiety of their own, often referred to as the biomolecular condensate. So although it's not surrounded by a membrane, it is a very open system. It can exchange with the surrounding cytoplasm, but the interactions that are occurring between these molecules and RNA in this small part of the cytoplasm allow them to stay together. And there are several interactions that actually contribute to that, right? There are cation pi, pi, dipole interaction that are happening. All of these are characterized by low affinity interactions, but they are made up by the number of interactions that occur, right? So there are lots of interactions of low affinity that lead to the assembly of these structures, right? And this is one of the fundamental bases of assembly of biomolecular condensates in the cells, including the P bodies and the stress granules that I was showing you in the previous slide. So this is how they assemble, and we are focusing on their disassembly part. And if you think about this, then it's a very interdisciplinary topic, because you apply, I mean, people who work on physics have talked about phase transition and phase separation for ages, right? They have looked at this in context of whatever they are working on. So it's a very old principle in polymer physics. Physical chemistry talks about it, but it's only now that biologists have started warming up to the idea. And although there are many biologists who poo-poo the idea of LLPS, and the joke in the field, in fact, goes that there are so many things phase separating that it will be now interesting to know what doesn't phase separate, right? Because biologists are now struggling to assign a function to what happens when you phase separate versus when you don't phase separate. But that's a discussion for another day. It doesn't change the fact that, yes, lots of biomolecules do undergo phase separation, and there is some evidence that that phase separation is tied to function, right? So it's a very interdisciplinary subject that we are trying to look at. Okay, coming back to my research, I said we focus on low complexity sequences, and here there are examples of some proteins that have these low complexity sequences, right? So you see that RGG is common to all these proteins. Along with that, they also have RNA binding domains like RRM, LSM, so on and so forth. So these are different sequences of proteins. All of them have low complexity regions. And when you look at their low complexity sequences, so I'm only showing you the part which has LCS, low complexity sequence, you will appreciate that what is there in the green are repeats of arginine and glycine. So we are very much excited about proteins that contain these RGG motifs. But if you take a close look, you will also appreciate that RGGs are also somewhat different in different sequences. Some cases you have RGG, in other cases you have RG. In some cases there are multiple continuous repeats of RGG, in other cases they are interspersed by phenylalanine, in some cases they are interspersed by tyrosine. So there is perhaps also some method to the madness, right? And one of the questions that I'm really interested in the lab is to try and understand what is the molecular differences between these low complexity sequences, right? Why are they all different? And we have evidence that they also have different functions. And that's the reason, as biologists, I'm interested in figuring out differences. So this is one aspect that I'm really looking forward to explore during this meeting. So if anybody is excited about similar questions, I'll be very happy to discuss. Okay, so all these proteins are RNA binding proteins, and they are components of RNP condensates. And in our lab, RNAs and RNA binding proteins come together and assemble to RNA granules, and it is known that these low complexity sequences containing proteins contribute to the assembly of RNA granules. This is already well known. But Raju, one of the PhD students who's now doing postdoc in James Shorter lab at UPenn, when he joined the lab, he started addressing the question, what about disassembly? I mean, these are dynamic structures, they form and disassemble. So what causes the disassembly of these structures? There are really no mechanisms out there that try and explain the disassembly processes of these structures. And that's something that I am interested. And like I said, he's already moved out, but his work that came out a couple of years ago provided one of the first instances of a protein that's involved in disassembly. That protein happens to be SBP1, and that disassembles P bodies through this RGG sequence, which is a low complexity sequence. So this was very surprising because low complexity sequences were already known to be involved in assembly. Raju went ahead and demonstrated that, look, LCS can also be involved in disassembly under the right conditions, right? So that was a very surprising finding that came out of his PhD work. And what that also did was to allow us to start asking questions which were not just relevant to yeast, but also in the context of certain specific diseases and in the context of human cells. So what is that question? So his work demonstrated that LCS could contribute to disassembly of physiological condensates. So P bodies and stress granules are physiological structures because they are there under normal conditions, right? They form and they disassemble and things like that. But there are also disease relevant pathological aggregates, right, which come up under the conditions of specific diseases. So Raju's work allowed us to ask that could these same things which are involved in disassembly of physiological condensates, such as the RGG motifs, can they be involved in disassembly of pathological aggregates, such as, for example, those which are involved in neurodegenerative disorders? So that's when we started asking this question, and which we have started doing in past 2 to 3 years. And Mani, who is now, he just submitted his thesis and will be defending next year, next week. And Naina, who is a second year PhD student, they both are collaborating very well to address this question in the context of neurodegeneration. And I'll be sharing some results along those lines. So we are now asking this question in the context of one specific disease known as ALS, or amyotrophic lateral sclerosis, also known as Lou Gehrig's disease, right? This is a muscle wasting disorder, and what happens as a result of that people who are unfortunate to have this disease, they end up losing all their mobility, and it's a motor neuron disease, and at some point they can't even breathe, and it's a fatal disease with no known cure for it, and as a result of that, within 2 years of detection of the disease, unfortunately the patients cease to survive. Okay, what is its connection to RNA granules or RNP condensates and things like that I was telling you in all this while? Okay, so it's a genetic disease. So there are mutations found in specific proteins that contribute to this disease, right? What are these proteins? The proteins such as TDP43, FUS, EWSR1, TAF15. If you again look at them closely, they have low complexity sequences. There are RGG motifs. This QGSY rich motif that's present in these proteins, they also are intrinsically disordered. Plus, they have RNA binding domains like RRMs and so on and so forth. So these are very much like the proteins that I was showing you earlier, which are from yeast, that they have low complexity sequences, they have RNA binding domains, and more importantly, they are also found to be present in structures which are like P bodies and stress granules, right? So how does that normally happen? It's very interesting because all these proteins that I was showing you in the previous slide, they are actually nuclear, right? So they're by and large present in the nucleus under normal conditions. However, in response to mutations and or stress conditions, so if I'm unlucky to be carrying some of these mutations, then as I grow older, then in the neurons of my brain, these proteins which otherwise would stay in the nucleus starts coming to the cytoplasm, right? So that is what is shown here. This is a neuron, by the way, and now they start making these aggregates in the cytoplasm, right? There is no nuclear signal. You can see lots of accumulation in the cytoplasm, and they resemble the condensates that I was talking about to the extent that they have the components of the condensates, but they are not dynamic. So they keep accumulating, and this is a feature of many neurodegenerative disorders, that having these plaques or pathological aggregates in the cytoplasm, which people believe are one of the causative reasons for the disease. So Mani and Naina are now asking the question that could we test if these low complexity sequences could reverse the process, right? So this is what happens in the disease. The nucleoprotein comes to the cytoplasm, forms condensates. Could these low complexity sequences make the aggregates go away and perhaps also see accumulation back in the nucleus? Okay, so what they did was they took this low complexity sequence from yeast and expressed it in human cells. That's why I'm calling these as yeastified human cells. You often hear about humanized yeast and humanized mice, but this is the reverse. We have taken a sequence from yeast, a protein from yeast, and expressing in humans. And we are asking, how does it affect the aggregation of ALS relevant proteins, right? And now remember, we are doing this in the context of human cells. Okay, so this is what Mani saw. Now you're looking at FUS. This is one of the proteins that's responsible for this disease. The wild type protein FUS is mostly present in the nucleus, so it doesn't come to the cytoplasm. Remember, I said you need to have some of these mutations which cause these proteins to come to the cytoplasm. If you don't have those mutations, you're fine, because FUS is an important protein, is present in all of us. But it's when there is a variant of FUS, that's when it comes to the cytoplasm. So wild type protein stays in the nucleus. There are no aggregates. You look at a mutation which is found in patients, which is P525L, you can now see there is very little nuclear signal, and you can see these aggregates that are accumulating in the cytoplasm, right? And this is blue color is representing the nucleus here, and you can see lots of aggregates outside the nucleus. What happens when we express this SBP1 from yeast, right? Again, wild type protein stays in the nucleus, nothing happens. What you're seeing in red color now, which was earlier sort of blank, is this yeast protein, which is actually being expressed in humans. We spend a lot of time. Is my time is up? Okay, all right. Expressing this protein. So that's 25 minutes, right? Or is it 20 minutes? 25. Oh, okay. All right. I better hurry up. And when it comes, when you look at the expression in the context of P525L, you can see that now these aggregates which are happening in these wild type protein is gone, right? These aggregates are not visible. That's the quantitation of what I'm showing here, that when you express this yeast protein, there is a significant reduction in the amount of FUS condensates that are present for the mutant protein. This is the control that says protein levels are not changing. We did the same experiment by enriching these condensates and then incubating them with either full-length protein or the RGG mutant version of this protein. And what Mani saw was something very interesting. They found that upon incubation with the buffer, there is a lot of protein that goes into the insoluble form, which is the aggregate form, and certain protein that stays in the soluble form. When you incubate it with SBP1, this phenotype reverses. Now you see less in the insoluble form, more in the soluble form, and this is the protein which it has been incubated with. Now you look at the RGG deletion mutant, where the entire protein is there, but the RGG motif is not there. The phenotype doesn't express itself, right? You again see it more in the insoluble form, like what was happening with the buffer, and less in the soluble form. So again, suggesting that this particular sequence is able to modulate these disease relevant aggregates and bringing them into the soluble form. Naina has looked at this directly, right? So Naina is very much interested into purifying proteins and putting it with these FUS condensates and asking what happens. So she has purified FUS protein, and she's allowing it to undergo in vitro sedimentation assay. So when you in vitro sediment it, you see very little of FUS in the supernatant and a lot of FUS in the pellet, right? This again recapitulates its behavior of the mutant outside the cell. The previous experiments were inside the cell, now it's outside the cell. This is when you incubate it with BSA, which is like a control protein. Now you incubate it with SBP1, you now see the reverse of this, right? There is a lot of protein in the supernatant, very little in the pellet, and this behavior is gone when you incubate it with the mutant that's lacking the RGG sequence. RGG is the low complexity sequence. So it's something that's dependent on the RGG sequence, that it now starts behaving just like control. So this behavior is lost. And this is, of course, quantitated here. This is the last data slide that I'll be showing. What now Mani did in this experiment was, we are switching from FUS to TDP43, which is another mutant which is well known in the context of this disease. He took just the peptide, right? All this while we were taking the full length protein. Now he has taken just the peptide, added it to the cells which are growing. So you're directly adding the cells to the peptide, and then he's looking at what is happening to these condensates, right? And so this is a TDP43 mutant, which is lacking the nuclear localization sequence. You incubate it with the ORGG sequence, and you look at it over a period of time, and what you see is the reduction in the size of this condensate, right? So this was at the 0-minute time point. This is what happens at the 60-minute time point. This is just adding the peptide to the media. You take a related RGG motif sequence, which is coming from another protein known as SCD6. This is one of the few RGG motif proteins that I introduced to you earlier. And this goes on to the idea that two RGG sequences are not the same, because this is also RGG motif. You take the same mutant, and what you find is that you incubate it, and there's not much change over the course of 1 hour, right? So this is 0 minutes, this is 60 minutes. Even though this is RGG rich, but the interspersed sequences are different, the number of repeats are different, and it doesn't behave like the SBP1 RGG sequence. This is the quantitation, this is the decrease in size for SBP1, whereas this is in case of SCD6. So what we are suggesting now is that the model of the and the moral of the story is that these low complexity sequences which affect physiological condensates looks like they could also be affecting pathological aggregates such as those of FUS and TDP43, which are involved in disease such as ALS. We are still trying to understand the mechanism of it, and a lot of this could be accelerated by doing molecular modeling and dynamics and things like that. So I'm very happy to discuss about this. But last but by no means the least, a big thank you to my lab, which I'm really blessed to have people who are very hardworking in the lab. It's because of their hard work that I get to go around wonderful places like this and come and talk about it. A big shout out to different funding agencies which has supported our work despite working on fundamental biology, and now slowly transitioning to somewhat more applied biology, and lots of collaborators all over the place. And a big thank you to all of you, including Sandeep and Debayan. I'll be very happy to take questions if there is time left.
So just one or two questions left to. Hello, very nice talk. So I had two questions. So first is it known that the RRGG motor, the peptide, it interacts with TDP or FUS there?
Yeah, so this result, I believe, is an in vitro
direct
system,
interaction?
right? So you only have two things here. One is the SBP1 and the other is the purified FUS. So the only way this could work is through a direct interaction. So yes, I think they interact.
And RRGG motif is known to interact with RNAs that you have talked.
Yes.
So does it make the entire complex more RNA interactive? And have you looked at the bigger size versus the smaller size condensate? Do they differ in
Behave
terms of
differently?
the behavior if you do
Yeah.
a PrAP or something on that?
Right. So those are some of the things that we are right now trying to do, where we are trying to add RNA to this. So this is a two-component system. We are trying to make it a multiple component system and see if you add RNA. We also have some interesting results with NAD and NADP, which we found earlier. So those permutation combinations, we are trying to look at it. Very relevant questions. I don't have an answer to that right now.
Yeah.
I have two questions.
Yeah.
One is that, is it always true or 100% true to say that RNP condensates always form through the liquid-liquid phase separation?
If I limit myself to structures such as P bodies, stress granules, P granules, there is enough evidence out there that suggests that it is a liquid-liquid phase separation. When you start going outside the gamut of these structures, because now LLPS is attributed not just in RNA biology but in many other cellular processes as well. So my knowledge of that may not be as good. So perhaps there are mechanisms other than LLPS contributing there. But are you referring to any specific structure which doesn't undergo LLPS?
Because sometimes I found that like the stress granules,
Yeah.
which are directly from the solid aggregates,
Uh-huh.
instead of making separated via the liquid-liquid phase separation.
Yes. So what you're talking about are more aberrant pathological structures, right? Yeah. So they do defy the norm, and that's the problem, right? That's why we have the disease, because it looks like they do not have the flexibility that the physiological structures have. Many a times the mutations and or stress conditions allow them to jump directly from one form to the sol or the gel form, right? And they are stuck in that form. So yes, that's possible, and there it often misses out on the LLPS part of it. Yeah,
So
that's
it
possible.
means the condensate's composition is very important for the liquid-liquid phase separation, right?
Including specific mutations,
Yeah.
because they affect these pi-pi, cation-pi, and dipole-dipole interactions. Yeah.
Okay. Thank you so much.
Yeah.